Yep.
Great. So good morning, everyone. We are here to talk about the fine-scale vegetation classification and map of the Modoc Plateau and surrounding region. As Rosie said, I'm Rochelle Bull, the lead vegetation ecologist with the Vegetation Classification and Mapping Program with the California Department of Fish and Wildlife. Most of you all know us as VegCamp. And VegCamp is the project manager for this project, although this project would not have been possible without many other people. We hope that you find the data from this project and the information presented today to be useful, and we'd love to hear your feedback after the day, but it's been brought to our attention that many people are unaware of the vegetation data that VegCamp supports, and so this is part of our effort to get the information and the data out to be used by the local managers and other potential users of vegetation data, because as you'll see, there's a lot of information and data here that could really drive and contribute to local land management decisions. Okay, so today I'll talk briefly about each component of the project and give you an overview of the process, as well as make you aware of the products and the data available that have come out of this project. So I'm going to talk about the scope of the project and the contributors. I'll talk briefly about what vegetation classification and mapping is, if you don't already know. I'll talk about the field sampling effort and the vegetation classification that came out of that. And then I'll talk a bit about the map itself, including the attributes that are embedded within it and where to find that data. I'll also show you the report that describes most of the things that I'll be showing you today and where to find it, at least in the near future. And then I will give you a quick project status update. So the scope of the project that we're going to focus on today is everything that falls within the Modoc Plateau and Northwest Basin and Range USDA defined ecoregions, which in this map is the brown polygons that you see here. So these ecoregions cover about 4.8 million acres, which is about 5% of the state. The project areas in green, teal, and pink cover approximately 47% of the Modoc Plateau ecoregion, which is this larger one here, and then about 37% of the Northwest Basin and Range ecoregion, which is further east. Field work for phase 1, which is in green, started in 2016, and phase 2 in teal started in 2018. And to define the phase 1 and 2 study areas, VegCamp relied up on the USDA ecological subsections. The boundary of phase 1 study area is about 1.1 million acres, you can see right there. And it excludes these large areas of private land that fall within the Madeline Plains and the Pitt River Valley subsections. And phase 2 of the study area is approximately 1 million acres and covers most of the Devil's Garden ecological subsection, along with all of Horsehead Mountain and the southern portion of the Aden Mountains and Valleys subsections. In addition, an additional 50,000 acres immediately adjacent to these, the phase 1 and phase 2, this footprint is in pink, and we call it the Applegate and Eagle Lake project area. It's encompassing the Eagle Lake Observation Peak and Bald Mountain Dixie Valley ecological subsections, and data collection started there in 2017. So overall, the study area is approximately 2.2 million acres, which leaves about half or 2.4 million acres of these two ecoregions for future mapping efforts. And then just for your information, we also have the Doyle-Loyalton mapping project that started in 2019, which is just to the south in orange. It's not fully shown here. This project is currently in progress and mostly outside the focal ecoregions and the scope of today's discussion, but just know that it's in progress. Okay. So although VegCamp facilitates the sampling, classification, and mapping of California natural communities, it's imperative that we work collaboratively with other programs, agencies, and contractors in order to move towards our goal of having a fine-scale vegetation classification and map of the state. And of course, this project was no exception. Phase 1 and 2 of this project, shown here now in all green, was initiated in 2016 with mitigation funding from Region 1 of the Department of Fish and Wildlife. And Amy Henderson specifically was instrumental in directing funds to this project to enable better conservation planning. So we'd like to thank you, Amy. And Amy, I know you're here. If you want to say hi, thank you. Or not. Hi, everyone. And then also the CDFW Wildlife Conservation Board also provided funding for this project under a grant. So phase 1 and 2 in green was sampled by field crews from the Geographic Information Center, or GIC, out of Cal State University Chico. VegCamp staff also did some sampling. GIC is also doing the mapping and the accuracy assessment. GIC, is anybody here from GIC? Brian Krebs or Jason Schwinkler? If you guys want to introduce yourselves. Anybody? Anybody looking at the participant list?
I'm on.
Yeah, hi, Brian Krebs here.
Jason's on.
And
Jason's on if you can hear me.
Yes, we hear you, Jason and Brian.
Okay.
Nice to hear your voices. Thanks for being here, and thank you for being so instrumental in this project.
Our pleasure.
All right, and then the Applegate and Eagle Lake in pink was funded by BLM, and sampling was done by the California Native Plant Society, and mapping was done and is currently being done by the Aerial Information Systems, or AIS, out of Redlands. Debbie, if I know you're here, I think John Menke's here too. Would you guys like to say hi? Introduce yourselves. No? You guys are being shy. All right, well, they're great mapping contractors as well as, and we've worked with them for a long time. And Julie from CMPS, would you like to say hi?
Hopefully I
heard you for a second.
Hopefully I'm not echoing anymore. Hi there, everyone. Thank you for attending this, and we're excited to be continuing to collaborate on fine-scale classification and mapping.
Great, thanks, Julie. And then again, the Doyle-Loyalton project, GIC is also mapping that, and started with funds from DFW's Big Game Funds. So we're very, very thankful and grateful for all the contributors and collaborators. All right, so now that we know the general region and scope and who we're working with here, I want to step back for a few minutes and just talk about what vegetation is, if you don't already. Vegetation communities are groups of species that tend to co-occur and repeat across the landscape. So what we're doing here is sampling these communities over and over again so that we can then name them and define them. And once we have clear definitions of the vegetation community or vegetation types, a map can be made, like shown on the right, that can be used. It's made in ArcGIS using aerial imagery, where the mappers will correlate what is being seen on the ground to what is seen in the imagery. So as you can see, it's really crucial to start with field data collection on the ground. But in order to implement a thorough field sampling effort over such a large geographic project area, it's crucial to develop a sample allocation to direct the sampling. And a sample allocation is pre-selecting samples before you go out into the field. And a good sample allocation will capture all the vegetation types within your study area and distribute your samples evenly across the study area, while also making sampling more efficient by reducing travel time and also reducing the potential for autocorrelation, which is basically when you have two samples that are too close to one another and are essentially within the same stand. So, for this project, Rosie Yakub, who you all heard her voice earlier, created the sample allocation by first limiting the scope of the allocation to only areas that are accessible to the field crews. Areas that are within a certain distance from a road, as well as properties that crews had permission to access. And then she used this schematic here of a random tessellation stratified survey design, or a GRITS design, to stratify the remainder of the study area. So speaking simply, she used GIS layers like these are examples here, like maximum and minimum temperature, precipitation, elevation, geology, or others, to come up with unique vegetation types should grow. So Rosie specifically for this project used the coarse CalVeg layer that was available for the project area, also the National Wetlands Inventory layer and the CAL FIRE layers to identify where these unique vegetation types should grow. And then she randomly generated points to direct the field crews to those areas. So this approach, in combination with manually photo-interpreted allocation points and also subjective identification of stands by field crews, was used to maximize the efficiencies and also increase the diversity of vegetation types that were sampled. So that sample allocation was then used to direct sampling, which, as I mentioned before, started in 2016 for this project. The CDFW CNPS Rapid Assessment Releve protocol was used to collect all the data for this project. This protocol includes a lot of information about the stand that you're sampling and its placement in the landscape, including some environmental data such as coordinate, slope, aspect, topography, soil cover, and much more. A general narrative description of the stand and any other interesting things about it. Information that tells us more about the wildlife habitat relationships, a field assessed vegetation type, and then strata, covers, and heights, and of course, species and cover values for those. It's important to note that while the sample allocation was used to direct the field sampling, the field crews played a crucial role in noticing repeating vegetation types that needed to be sampled. So perhaps the sample allocation was repeatedly missing a certain vegetation community, or the allocation points were placed in like stand transitions. It was the field crew's job to make sure that all community types were being captured and that the surveys were being placed within homogeneous sites. You can read more about the protocol for this data collection by following the link at the bottom here, or by going to the VegCamp website and going to the Publications, Protocols, and Standards page. The form and sampling protocol are also appendices in the report for this project that we will talk more about in a few minutes. So the goal for data collection is always to collect data evenly distributed across the study area and within all possible vegetation types. So here's the distribution of the survey points collected within the project boundaries. The survey points are available actually through the Biogeographic Information and Observation System, or otherwise known as BIOS, which is a data viewer maintained by CDFW. I can talk a little bit, I'm going to talk a little bit more about BIOS later, and I believe Rosie will too. So but just know that this survey data is available for you to view and download from BIOS. Once we have all the field data collected, we then analyzed the data. VegCamp ecologists analyzed the data to develop our vegetation classification for the region. So in total, you can see down here at the very bottom, GIC and VegCamp combined for phase 1 and 2 of the study areas collected 627 surveys. CMPS collected an additional 230 surveys within the Applegate, Eagle Lake areas. In addition, another nearly 1,200 surveys, existing surveys from other projects and agencies were gathered to be co-analyzed with the data collected to create the more comprehensive vegetation classification. The other data sets that were included were U.S. Forest Service ecoplots that were collected on the Modoc National Forest in the late 1980s and early 1990s, as well as recently collected assessment, inventory, and monitoring, or AIM surveys collected by NRCS and BLM that was sampled within the focal ecoregions, and then also included were samples from the U.S. National Park Service for the Lava Beds National Monument. That's this big dark area up here with all those points. And then also some VegCamp surveys from past projects. So in total, there were over 2,000 surveys included in this data analysis. All this data was quality checked and standardized and prepared for data analysis to create the vegetation classification that defines the types within the ecoregion. So the vegetation classification analysis entails grouping similar plots together based on species covers and abundance. So this colorful spreadsheet in the middle that I don't want you to read is showing an example of conifer types, each line, conifer surveys. So each line is a survey that are grouping together, and each color ended up being a different community type. And the fact that these communities are grouping near each other also indicates that they're related to each other. So all this information goes into creating a hierarchical vegetation classification that follows the National Vegetation Classification System. If I zoom in here to a part of the hierarchy, you can see the different levels of the hierarchy, from class level at the very top all the way down to association level. The finest levels of the classification are the alliance and association levels here, with the associations being listed on nesting under the alliances. Above the alliance and associations are broader levels that group communities that are geographically and ecologically related. So the hierarchy shows the relationship between communities. And as I mentioned, the final classification follows the National Vegetation Classification System, which allows us to maintain a national and international perspective and gives us the ability to communicate across state boundaries in regards to these communities and their relationship to wildlife. Of course, we also maintain a local and state perspective that will highlight and represent California's unique communities. So this hierarchy can be found as Appendix A in the classification report that I'll show you in a little bit. Let's see. So a little bit more about the hierarchy. It's important for many reasons. Some of them I just explained, but Also, one reason being that it demonstrates how community types are related. So for example, this Artemisia tridentata, or Big Sagebrush Alliance, over on the left, and on the right we have an Eriogonum poecunda Alliance on the right. They fall within the same macro group here. They fall within the same class, subclass, formation, division, and macro group. The macro group is intermontane, tall, and dwarf sagebrush scrub steppe macro group, which tells us that they're both found in the intermontane western US, where the landscape is dominated by sagebrush communities, but then they fall under different groups that are under this macro group, indicating then the Eriogonum falls under the Great Basin intermontane dwarf shrub steppe group, and the Big Sagebrush, surprise, it's under the intermontane big sagebrush steppe and shrub group. So this indicates that the Eriogonum Alliance is more related to the dwarf shrub or dwarf sagebrush group community types, low sagebrush. So for this project, 83 associations that fall within under 61 alliances were described for the area. 4 new alliances and 28 new associations for the state came out of this analysis and are likely mostly restricted to this northeastern corner of the state. In addition, 26 of the described alliances and 48 of the associations are considered sensitive natural communities in California. For example, this photo shows a newly described vernal pool alliance from this project. These pools have a different suite of species than what is found in the Central Valley vernal pools. Species such as this here, Downingia botchagluppii. It's a vernal pool specialist that is, for the most part, only found in these ecoregions of California. So from these, each of the alliances and associations, there's descriptions that are created, which can be found in the classification report as Appendix J. I don't expect you to read all of this, but this is an example of the Western juniper description that can be found in the report. In here, you'll find a general alliance concept for the type, a local distribution, a list of the associations under the alliance found in the study area, summarized environmental information, and then this thing that we call the stand table. The stand table summarizes what species tend to occur in this alliance, the constancy of each species, the average, minimum, and maximum covers for each species. And just to clarify, the constancy right here, CON, at the top, is the percentage of the surveys that a species occurs in for a particular vegetation type. So, for example, I'm going to actually use this highlighter. Let's see, okay, it's on yellow. Okay, you can see here that Persia tridentata, you may, or hopefully you can see, is right there. It's in 53% of these stands. That's what the constancy means. So there's 343 juniper stands that went into defining the alliance for the study area. 53% of those have Persia tridentata present at about 4% cover in those 53%, in those plots. So that's how you use stand tables. And those descriptions can be found in the report. So in addition, I'm going to take off my highlighter so I don't highlight everything. In addition to the descriptions, there's also that the all that information is summarized into a vegetation key to each alliance and association. This key is then adapted to be used for determining the vegetation types in the map, as well as it is used when determining vegetation types in the field. So any of you could take this key into the field and determine the alliance and association that you're in. So all of those components are the things that come out of the vegetation classification analysis. So now I would like to move into talking about the vegetation map. Of course, the vegetation classification and the rules that drive the classification also drive the vegetation map. So the goal with the vegetation map is to create a fine scale or alliance and association or association level map that is wall to wall and finely divided. So where each polygon contains a lot of information. So if you zoom in to this map and look at an individual polygon, say this polygon here, there it is, and open up the attribute table for this polygon, you'll see that all the information that is embedded in this map for each and every polygon in the vegetation map. Here's the list of attributes. Of course, you have the vegetation type at the top that is based on the classification that was created from the data collection within the region. And as I said, this is mostly an association level map. This is the Juniperus occidentalis, Artemisia tridentata, Persia tridentata association. That's what this map, this polygon is attributed as. You have the strata covers, so tree, shrub, and herb, and total vegetation cover for each polygon. You also have the height of the dominant vegetation layer and the tree size class if there's trees. There's a few project-specific attributes in regards to the expansion of Western juniper within the region. You have presence of isolated trees to help identify roosting and nesting sites for raptors and owls. Attributes for various disturbance to the vegetation. Comments and other fields that give you more information about the specific polygon. You have the vegetation hierarchy embedded in the map for each polygon to provide flexibility for the users. Then there's a crosswalk to other classification systems like CAL and CWHR or wildlife habitat relationships. And then there's the rarity rank is also listed for the finest level of the hierarchy that is listed for the polygon. So let me go into a few of these attributes a little bit more. I've already talked a lot about the vegetation type and how it's derived, but something that's related that I like to highlight is the hierarchy. As I mentioned before, the classification is hierarchical, which means it's scalable. And this feature, as I said, is embedded into the vegetation map. So this allows the users to scale the map to the level that is most useful for their purposes. For example, this map here is showing the landscape divided into the finest level available in the map. So this is association. This is showing several juniper associations in various shades of blue. We have some low sagebrush, some big sagebrush, bitterbrush and sagebrush, and then like a tiny bit of grassland there. So this level can be useful for identifying, say, like potential rare species habitat or sensitive natural communities, and also for directing local level management. But if perhaps you only need to use the data at a broader level, the alliance level, say, because maybe you want to identify the location of all the juniper woodlands. So now we've merged all the juniper associations into the juniper alliance. So now you can see that this simplifies the map considerably. And so you can use that to direct management more broadly. Okay, next I'd like to highlight the strata cover attributes. So for each polygon, a percent cover is estimated for the total tree layer, and also separately for conifer and hardwood covers. And then of course they have the shrub cover, the herbaceous cover, which is in a cover class, and then the total vegetation cover. So this polygon has 0 hardwood cover, 16% conifer, probably all juniper, and therefore 16% tree cover, 4% shrub, and then somewhere between 10 and 40% herb, which all adds up to 45% total cover. So in addition to dividing the polygons on vegetation type, the polygons are also divided based on these strata covers. So if you look at this map here on the left, you'll see that all the blue polygons are a single vegetation type. They're all the Juniperus occidentalis, Artemisia tridentata, Persia tridentata association. So it's the Western juniper, big brush, big sagebrush, antelope, bitterbrush association. And the associations, you can see that these are all the same association, and you can see that there's polygons that are right adjacent to each other. So rather than being divided by vegetation type, these are being divided by conifer or shrub cover. And if you look at these two photos over here, which are both taken within this same association, but different stands, you'll see that they look very different. The one on the left shows a more open juniper woodland with fairly high shrub cover. Excuse me. And the one on the right has much higher cover juniper with much lower and much a lower understory component. So the differentiation of stands based on strata cover can provide more direction for management depending on your goals. The next set of attributes that I'd like to highlight for you is these two attributes here, the juniper expansion and the restoration attributes. These attributes are particular to this mapping project. For all mapping projects, as part of our process to involve and consult with the local land managers within the region to determine what's important information for them. So what, in addition to our typical attributes, would be useful for them to make better and more informed management decisions. And these attributes came out of that discussion. So I'm sure I'm speaking to the choir here, and you guys mostly know this, but the range of Western juniper has expanded in the last 150 years due to fire suppression, overgrazing, and climate change. And this can impact grazing habitat for deer and livestock. So the juniper expansion attribute identifies polygons where young junipers have expanded into shrubby or herbaceous landscapes. Attribute also has an intensity range that identifies when there's minimal, moderate, or high expansion of young junipers. So that can be used as, thresholds of when removal of juniper or restoration might occur. And the restoration attribute, identifies sites where juniper removal has already occurred, so that those stands and those communities of those stands could potentially be tracked over time to see if that restoration is moving in the direction that is best for the landscape. Okay. I know this is a lot of information. So let's talk a little bit about disturbances, these disturbance attributes. Within each polygon, the presence and severity of disturbance from clearing, rodentness, development, and invasive plant species is noted. So Rosie will talk more in depth about the possible uses of at least one of these a little bit later, but in general, using this attribute, you can differentiate between highly disturbed and relatively pristine sites, and then spatially identify them. And then in combination with other attributes, you can determine potentially areas of potential restoration or conservation. So here's an example of a clearing of, this would be high disturbance from a clearing. This is invasive species. This is a grassland being invaded by non-native exotic grasses. This here is showing an example of high rodentness. This would be pretty low rodentness. This is a really beautiful picture of an example of development. And I have to say that a lot of the photos from this PowerPoint are from Brian Krebs with GIC, who takes some beautiful photos. And also Todd Keeler-Wolf provided many of these photos, in addition to a lot of our just field photos that we take during field sampling. Anyway, so that's disturbances. And okay, the last attribute I want to talk about is rarity, this rarity attribute. So for each polygon, the global and state rarity rank is listed for the finest level of the hierarchy that is attributed for the polygon. So this polygon is attributed to the association level, where we give yes and no, yes/no rarity ranks. And you can see here that this association is a no, so which means it's not a sensitive natural community. Diana Hickson will, I think, for me, she'll be talking a bit more in depth about the rarity ranking. So I don't want to go too far into it, but I do mostly want to point out that, again, this attribute is embedded within the vegetation map for each polygon so that you can spatially identify the sensitive natural communities. And also I want to point out that Appendix A in the report lists all the vegetation types that are found within the study area, along with their rarity ranks for the alliance and association levels. Okay. All right, an important part of following the mapping standards that VegCamp has developed is testing the map accuracy and reporting the accuracy for each community type mapped. The accuracy of the mapped vegetation types is done through a formal accuracy assessment where field crews assess specifically allocated polygons without knowing what the mappers called it, and the field data is compared to the map types, and a score is given for each polygon assessed. Then these individual polygon scores are then summarized to give an overall score for the map and a score for each type that was mapped. So here's our polygon that we've been looking at this whole time. This point here is an accuracy assessment of this polygon. And you can see here in yellow, these pieces of data that have yellow blocks around them are the data that was collected in the field for this accuracy assessment. You can see that they called it juniper with big sagebrush and bitterbrush. They gave strata cover estimates in the field, as well as species cover, and some general comments on how much of the polygon they've used. The pink cells here is the photo interpreted, or the mappers call for the vegetation type and strata covers. And then the blue cells indicate the final vegetation type, score, and other notes determined by VegCamp ecologists for the final assessment and the final call. So I'm not sure if you can see, but like I said, the field crew called it Juniper Artemisia tridentata, Persia tridentata association. The mappers also called it that, and the final assessment by the veg camp ecologists agreed. So the mappers got this 100% correct, which each score is out of 5. So they got a 5 out of 5. And then we do that for several hundred of the polygons to determine the accuracy of each type and for the map. I do want to point out that the comprehensive accuracy assessment results will be included in the final mapping report, not this classification report. That mapping report will be due in the spring of 2021 along with the methodologies used. But the preliminary accuracy assessment result for phase 1 of this project can be found in the classification report as Appendix I. So I keep talking about this report. Everything I just described except for the mapping information can be found in this classification report. The report can ultimately be found through this link. It isn't there quite yet, but it will be soon. I'd like to quickly go through the report with you. I have a draft report right here that I'm going to just open up, and I'm going to just show you these things in the report that I just went through. So bear with me. Let me know if you don't see something, if you don't see it in a second. How's that? I know it's kind of small. Here's the report. There's Amy Henderson doing field work. You see it?
Yep.
Okay, great. So again, in the report, you have the project design and who funded it and everything, the background for this project. You have the study area description. Here we go, off the page there. And then, of course, we have more in-depth methods for the sample allocation for this project, and also the field sampling and data analysis. And then of course, you have the results that include information about the taxa that were included, and then the classification that came out of that, and then a little blurb about the vegetation map. But that's mostly going to be in a report yet to come. So the products that I showed you all are mostly appendices in this report. Appendix A being the vegetation. I feel like we're getting cut off here. Sorry, is that big enough? The hierarchy, the classification and the hierarchy, which also lists the rarity ranks for the alliance and association level, and whether or not the type is newly described based on this project, or if it's a range extension. Many of them were existing types as well. You have Appendix B, which is that field form, the rapid assessment releve field form that I mentioned, as well as the protocol, everything you'd want to know about how to do that survey in here. The plant taxa included in the data analysis and how we dealt with each of those things. An example of that cluster analysis, or the dendrogram that I briefly showed you guys. There's also this table that shows noteworthy or basically rare species that were found within the surveys sampled for this project and what vegetation type they were found in. Here's the key to the vegetation types. This is broken up by life form to make it a little easier. So tree, shrub, and herb sections of the key. This classification crosswalk to other classification systems like CWHR and CalVeg. Remember, this is also embedded in the map for each polygon. That accuracy assessment, preliminary results that I mentioned, and then at the very end, last but definitely not least, there's the vegetation type descriptions that I mentioned. Again, broken up by tree communities, shrub communities, and herbaceous communities right there. you can go to each one of these, say the AB's Concolor or White Fur Alliance. There's a nice picture in there, has all the description that I described to you, and classification confidence, maybe the sample size that went into describing this alliance for the project area, and that stand table. And then you can, the same information is listed for each association under the alliances too. Okay, so again, that I think we're going to show you where we store all our reports on our website. Ultimately, this link will work for that report, but it doesn't quite yet. Okay, I have just 2 more slides and then I swear I'm done. So I want to give you a quick status report on these projects. Phase 1 here in dark red is 100% complete and can be found on BIOS. Phase 2 and Eagle Lake maps are in pink here and are scheduled for completion next spring of 2021. And also, just for your information, the Doyle-Loyalton vegetation map south of the project areas, we've discussed it earlier today. It's still in progress. Its completion is yet to be determined and is contingent upon additional funding, although all the field data sampling has been completed, the data analysis is complete, and some of the mapping is done. We just need to finish mapping and do an accuracy assessment for that. Again, I keep mentioning BIOS. Here's what BIOS looks like. I think Rosie will be doing a live demonstration of BIOS, but just so you know, here are those 3 data sets that I mentioned. There's the phase 1, which in BIOS we call the Modoc Plateau, Shin, Likely, and Snowstorm Mountains, which is DS2877. Here's the Applegate vegetation map that is complete. It's DS2858. And then here's that survey points layer that I told you about, DS1020, which has all the survey points that were collected within this project area, as well as throughout the entire state. So you can also type vegetation up here and it'll get you a list of other veg maps that we support. I think that is all I have for you guys, and I know it's a lot of information. If you guys have questions, Betsy's monitoring those. Betsy Harbert, she works for Veg Camp. She's monitoring those, and we will answer questions, I think, at a break. So I think right now, Diana Hickson is going to start.
So, Rochelle, we do have a few minutes to answer a couple of questions.
Okay, yeah, let's do it.
Yep.
Who has?
We do have 2 questions already in the chat, but if you guys want to submit more, you're welcome to. So the first question is from Denny Grossman. Are there G ranks and S ranks for each association and alliance?
For California, our associations, Diana's going to go more into this, so I don't want to go too much into it, but the GNS ranks are listed for our alliances. I don't know, we don't actually give global ranks, but NatureServe does, and so if they haven't ranked them, then they don't have global ranks. But we do try to rank all of our alliances for the state. And then the associations, it's a goal to rank them all, but as of right now, most of them are not given official G and S ranks, but more of a generalized yes, no for rarity. But those all have a yes or no listed, and some of them are ranked. So hopefully Diana can talk a little bit more about that, and that'll clarify a little later or next. What's else?
Thanks. The next one is from Laura Snell. How will this data be updated? Is there long-term funding?
how will the data be updated? So like repeat mapping, like
I'm assuming
map update?
remapping, maybe. But Laura, if you want to clarify in the chat, you're welcome to.
Or if you want to just talk, you can. Laura. Well, I mean, it's a good question. Funding isn't secured for remapping if we're talking about remapping, but you know, it's good to get the baseline in order to be ready for remaps. And of course, if there's local funding that would support that or additional funding beyond, you know, hopefully we'll be ready for that and have the baseline map. So that's
It's definitely our goal to remap, and some areas of the state have been remapped, including the Central Valley, Western Riverside County, and Sassoon Marsh.
the Delta.
And the Delta. But it depends on funding right now. Eventually, we hope to get stable funding to be able to remap portions of the state based on the amount of change that's occurred or, you know, demands from users.
Thank you, Diana.
Yeah, and in the cases of the places that have been remapped, it's really been kind of local or specific interests that have made sure that it happened. You know, for example, Sassoon Marsh, it was part of the general management agreement that there would be remapping done there. DWR had an interest in mapping the Great Valley, and so they made sure that that was remapped. So usually it's a more local instance that drives remapping.
Yeah. Thank you, Rosie.